we discussed the mechanism of RNA polymerase transcription. So once you have the promoter, all the factors are assembled, mRNA starts to synthesize. As soon as mRNA is about 20, 25 nucleotides, just beginning, right? It starts to get capped at the 5' end. So capping adds some layer of stability while it is correct, still transcribing the rest of the gene. Does it make sense? And then elongation, termination, polyadenylation. Polyadenylation is done by a different enzyme, not by polymerase, not by RNA polymerase. There is a different enzyme called poly-A polymerase, which adds the poly-A tail, is about 100 to 200 nucleotide long. Right? That is again for stability and export of the mRNA from nucleus to cytoplasm for translation. So read this. Let's see, we will come back to the regulation today. Okay, so let's go directly into the prokaryotic gene regulation. So transcription, what we discussed so far is how exactly transcription is initiated, elongated and terminated. Correct? The basic mechanism, how it happens in prokaryote, how it happens in eukaryote, the promoter structure, the transcription factor, the enzyme. But transcription must have to be highly regulated because you don't want to keep on transcribing an mRNA if there is no need. So when there is a need, you need to transcribe the mRNA so that it can make protein and does the job. And when you don't need the transcription function, you shut it down. Correct? So one is called activation, another is called repression of the gene. So this has to be regulated, and we will give some classic examples how the protein coding genes are regulated in prokaryotes. Eukaryotes is much more complex. It's getting better in terms of understanding, but it's like a ocean how the chromosomes are regulated. So in prokaryotes, genes for enzymes for pathways are grouped into clusters. What it means is the polycystonic nature of the prokaryotic genome organization, correct? That you have a protein coding, you need maybe 5 different enzymes for a particular pathway, and all the enzymes are coded in the same region and they're controlled by the same promoter. So that whenever this pathway needs to be turned on, all the proteins, all the genes are transcribed at the same time into corresponding messenger, single messenger RNA, right? And when this is transcribed, translated, you actually produce 3 different proteins or enzymes. So multiple genes are coded in the same open reading frame. This is called ORF, open reading frame, or commonly transcription biologists like to call it ORF. So this is the promoter, this is the ORF. In this case, the ORF codes multiple genes. Does it make sense? And this happens primarily in prokaryotes. So when it is transcribed, whole open reading frame is transcribed into a single messenger RNA, and that messenger RNA, when it is translated, you produce different enzymes. Does it make sense? So polycystonic, and this is what it is telling, that genes for enzymes for pathways are grouped in clusters on the chromosome, and these are called operons. Operons means whole thing, whole gene, genomic structure, including the promoter regulatory region, whole thing is called an operon. Normally you don't see here the operon system in eukaryotes, only in prokaryotes. Operon means operatory system including the gene, correct? So this is operon. This allows the coordinated expression. A regulatory sequence adjacent to such a unit determines whether it is transcribed. This is called operator. We are going to talk about it. Regulatory proteins work with operators to control the transcription of the protein coding genes. So there are many such operators, operons in eukaryote, I mean the prokaryote, like lactose metabolism. There are a couple of genes, enzymes which are required for lactose metabolism, and so these enzymes and its regulatory system is called lac operon. We will talk about. There is something called tryptophan, TRP operon, galactose operon. So different metabolic pathway has different cluster of genes and regulations. So if there is a lactose metabolism has to be turned on, that means cells are treated with lactose. Correct? It needs energy, so it hydrolyzes the lactose, and so you need to turn on that operon so that enzymes are produced. We'll get into the lac operon in detail in a minute. So there is two terms, one is called induction and another is called repression. Induction means the gene is there. Normally this gene is silent, it's not expressing RNA. Okay, that's normal situation. But when there is a need, you turn on this gene so that you make mRNA, and this is called gene activation, or this is called induction. What it does is that within short time frame, it makes more RNA molecules. That's induction. If it is repression, that process is very slow. More RNA means, where do you think is regulated? How does it produce more RNA? What is the mechanism? Yeah, that's the regulation of the protein phosphorylation, dephosphorylation. But what happens to make more RNA? You need more initiation event. Does it make sense? So you, within short time, you do the more initiation so that it can make more RNA. Does it make sense? So induction or activation of genes actually boils down to more transcription initiation events. Frequency of initiation events is more, correct? So that's called induction. At the same time, repression is just opposite. Some gene is there, you are inhibiting the expression of this enzyme, that's called repression or silencing. Increased synthesis of genes in response to a metabolite is induction. Decreased synthesis in response to a metabolite is repression. Some substrate induce enzyme synthesis. Correct? When there is a need of glycolysis, you need to turn on the first enzyme, which is, what is the first enzyme in glycolysis? Hexokinase. So you need to turn on the hexokinase, correct? In response to low glucose, low energy of the cell, low ATP, correct? Or increased ADP, high adenosine diphosphate, less ATP, activates this particular gene or triggers the glycolysis, correct? So ADP is a metabolite or second messenger. It involves activation of the hexokinase family of genes. Does it make sense? So that's an example, like citric acid, high citric acid. Correct? It's high energy for the cell. Correct? So you want to slow down some of the glycolysis. So that is a repression. In eukaryotes, insulin, correct, triggers glycolysis. Insulin will increase the rate of glycolysis. Epinephrine, correct, will increase the glycolysis only in the heart muscle, while it inhibits in the skeletal muscle. So those are differential regulation of the gene by the metabolite, hormone, nutrient, salt, temperature, correct? All this stuff. So these are called inducers or repressor depending on the mechanism of action. The one of the very well studied operon in eukaryotes, in prokaryotes, is called lac operon. So lactose you have studied, correct? Lactose you have studied. What is the composition of the lactose? What are the two? It's a disaccharide, correct?
Yeah.
What are the two disaccharides? Galactose and glucose, correct? So what is the linkage here? Can you read? One what? Alpha or beta? This is beta. Why beta? Same side of the CH2H, correct? Same case. That's beta. So that's why it is called beta D-galactopyranosyl. What is pyranosyl? Why is it called pyran? 6-member ring with oxygen is called pyran. Correct? We have studied all this. Right now we are very small. Correct? So this is beta D-galactopyranosyl 1,4 beta D-glucopyranosyl. Is it alpha or beta? In this case it is beta, correct? Is it reducing or non-reducing? Why? This OH is free, correct? So it's a reducing, you are right. So once you hydrolyze this particular glycosidic linkage, you need some, correct, galactosidase type of enzymes which will hydrolyze this into glucose and galactose, and that can be metabolized. So lactose needs to be metabolized, and it is controlled by a system called lac operon. I want you to know at least this one in detail. Lac operon. This is basically metabolic system, regulatory system for lactose metabolism. You need an enzyme which will hydrolyze the glycosidic linkage between galactose and glucose, correct? That particular enzyme is actually beta galactosidase. This gene is called LAC-G, lactose metabolism related gene. That's why it is called LAC, correct? G. It also has some other protein, some other enzyme which is called LAC-Y and LAC-A. All of these enzymes are required for lactose metabolism. So all these three enzymes, LAC-G, LAC-Y and LAC-A, correct? LAC stands for lactose metabolism. Does it make sense? These are fused together as a single open-reading transcription open-reading frame, and it is regulated by a promoter. In this case it is called LAC operator, T-LAC O for operator. Operator means it's a regulatory sequence which controls the expression of the operator is nothing other than the promoter sequence where your polymerase etc. bind. Does it make sense? So this is LAC operator, and this activity of this LAC operator is controlled by something called LAC repressor. There is a protein which actually controls the activity of this LAC operator, and this protein called LAC repressor, and the gene coded by the LAC repressor is called LAC-I. So LAC-I, if it is transcribed, it produces LAC repressor. Does it make sense? So this LAC repressor is controlled by its own promoter, which is here. So there are few things. LAC-G is the beta galactosidase, LAC-Y and LAC-A, all three are involved in glucose lactose metabolism. These are controlled by operator, and operator activity is controlled by LAC repressor, which is coded by LAC-I, and LAC-I expression is controlled by its own promoter. Now, this is the mechanism how LAC repressor controls the LAC operator. So LAC repressor, LAC-I gene codes for LAC repressor, so when it has its own promoter. When LAC-I is transcribed, you produce LAC-I specific messenger RNA, which is translated into the corresponding protein, which is called LAC repressor. Does it make sense? And this LAC repressor normally forms a tetrameric structure, homotetramer. When this LAC repressor forms tetramer, this tetrameric LAC repressor goes and binds to the LAC operator sequence. Okay, this lock, and this is the promoter of the LAC operon. So this operator sequence is bound by the LAC repressor. Once the operator is occupied by the LAC repressor, it's a repressor, correct? It does not allow the gene expression. Does it make sense? So gene is repressed. Normally, the LAC repressor is bound to the operator so that promoter is not active, so it cannot express these genes. So if it needs these genes to be expressed, this LAC repressor has to be dissociated from the operator. Does it make sense? And this is exactly happens that with a inducer, there are certain inducer which can bind to this tetrameric LAC repressor. So LAC-I is transcribed into corresponding messenger RNA and then into LAC repressor, which forms tetramer. And inducers are some kind of molecules, it's kind of lactose type of molecules. It binds to the LAC repressor tetrameric form. So once this LAC repressor is bound to this inducer, this LAC repressor cannot bind to the operator. If it cannot bind to the operator, your promoter is open. Does it make sense? It now transcribed. Is it clear? So this is a very well studied classical example of transcriptional regulation using LAC repressor. If you go to another operon, TRP operon, tryptophan metabolism, similar type of course there are other proteins involved, but this is the basic mechanism. And once LAC repressor cannot bind, it transcribes the messenger RNA, all the genes together, and when it is translated, we form three different proteins. Does it make sense? So this system has been widely used in recombinant technology to express any gene of interest. So this is native, okay, in the cell, but you can clone some of the genes in this open reading frame. Instead of LAC-G, LAC-I, or any gene you put here, it can be regulated by this promoter. Okay, so let's say you want to produce recombinant insulin. Insulin is so important. correct? Lot of people have genetic history of diabetes, epigenetic, correct? Influence on type 2 diabetes, so insulin is needed to treat this patient. So you need the recombinant insulin protein. So what you can do, you can clone this insulin gene in a vector which has the lack operon. Does it make sense? Now you transfect these in E. coli, correct? And then put the inducer. Does it make sense? So now, because inducer will activate this lac operon, you will produce insulin. Does it make sense? So you can make kilograms of insulin and give to the people who needs it. But that is not so successful. Although you can express a lot of insulin, this insulin is not good for, not so good for treating diabetes. Why? This insulin is not as useful. You are not wrong. That means there is a better answer. Yes. Anybody else? Little more advanced biochemistry. Why this insulin is not active? The insulin is not active. It's not active. If you inject this insulin in human, it's not so active. Why? What is that? Proinsulin. Proinsulin. Let's say you cleave and give, still it does not work.
It will not bind to receptors on human cells?
You are not wrong. But what is the, yes?
Is it just because it doesn't go through all the post-translational modification?
Post-translational modification. Correct? It's a eukaryotic protein, eukaryotic hormone, and once you express in prokaryotes, prokaryotes do not have the post-translational modification machinery. So yes, it does not go through post-translational modification, so it does not have the right conformation, right? Correct? It does not bind to the receptor so easily. So that's why it is activity is low. Does it make sense? Whenever you are trying to do any protein work, any enzyme function or treatment in eukaryotic organism like human, you have to make sure that you express this protein in a eukaryotic system. Normally you could express these in fly. There are fly cell lines where instead of E. coli, you use fly cell line. Okay? So you could actually express this, then it has some better activity. Many times, lot of biochemists, correct? Traditional biochemists like to study the single enzyme function in kinetics and so on and so on. But one of the major problems they face or the kinetic data, what you get by expressing the protein in E. coli does not match with the in vivo function. And that's one of the reasons that because the eukaryotic protein was expressed in prokaryotic system, it does not have the proper modification, proper function. Does it make sense? So if you want to study a eukaryotic protein, either you can directly isolate the original protein from the human cell or eukaryotic cell, or express the recombinant protein in a eukaryotic organism so that you have higher chance of proper conformation, folding, binding, and so on. Does it make sense? So this is one of the technology which people use, take advantage of the lag repressor to express the protein in prokaryotes. Lot of time crystal structure you need lot of protein. You express in E. coli, and E. coli can grow as much as you want, purify, correct, and work. So lac operon is a very classic operon where you can use lac repressor to control the lactose metabolism related gene. So simple, it has a lac repressor which is coded by the promoter. It is expressed into RNA protein form a tetramer. And in the absence of any inducer, it binds to the operator. But as soon as you have a inducer bound to the tetrameric lac repressor, it cannot bind to the operator. Why? In presence of inducer, it cannot bind to the operator. Why? There is a conformational change. Correct? It has to bind to the DNA. So DNA binding conformation has to be right so that it can bind. So if there is a conformational change, it cannot bind more efficiently. So now promoter is open to transcribe. We transcribe all three genes as a single mRNA and then translate it into three different proteins. Yes?
So the repressor is always expressed, it's just modified to single.
Exactly. So the repressor should be expressed. It should be bound. Correct? But whenever you need, you can turn on the operon by inhibiting the repressor. So basically you are inhibiting the repressor. And one of the classic inducer is called isopropyl beta thiogalactoside IPTG. Any of you work in a biochemistry lab to express protein? You use IPTG, correct? You transform with the plasmid, grow it, and add the IPTG. Keep it for 2-3 hours. You see the protein expression is going on. So IPTG is an inducer for lac operon. That's what they need to. So this is the general organization of the lac operon. Operon consists of transcriptional control region and a set of related structural genes, all organized in a contiguous linear array along the chromosome. Correct? Transcriptional control regions are the promoter and the operator, which is next to overlap upstream from the structural genes they control. Correct? And then operators, whatever we discussed. So in your exam, I can ask, correct? Draw a lac operon and tell me or briefly explain how it is regulated by the lac repressor. Does it make sense? Yes or no? Can I ask? Very feeble. Can I ask that question? Yeah, that's good. If you tell me what question to be asked, I'll ask. Okay? There are other regulatory stuff. I'm not getting into this. Catabolite activator protein called CAP, which is also another regulator of the protein, so I am not going to get into detail. So that's the lac repressor in the prokaryotes. This is, I'm not asking you to know all of it, it's called ERA BAD operon. That means it is involved in arabinose metabolism. TRP operon. This is involving tryptophan metabolism. There is again like lab repressor, you have ERT repressor. Does it make sense? So these are similar, these are well studied, and I am not getting into detail. In fact, if you are studying this, then you need to go into detail. But as long as you understand the lack of operon, how at least one operon is controlled in prokaryotes, my job is done. Okay, so there are a lot more complex regulations. Let's keep all this stuff. Now we talk a little bit regulation in eukaryotes. That's what my research is. How the genes are regulated, turned on, turned off in eukaryotes, especially in human system. Okay, so in prokaryotes, your DNA is relatively open because there is not much. Correct? Histones, there is no histones, there is no nucleus, so your DNA is not totally free double-stranded DNA. There are some DNA binding proteins, some secondary structure, some folding, but it's not like eukaryote. In eukaryote, your DNA is compact inside the nucleus, correct? There are variety of complexations, and one of the simplest structure where DNA can be seen is called nucleosome. Are you guys familiar with nucleosome? You did in Biochem 1? Let's see if I have some slides. This is part of my research talk. You don't have to know in detail, but I think it's good to know. Correct? My lab studies the epigenetics of human gene regulation. That's what we are going to talk now, correct? Steroid hormone signaling. Steroids are critical for a variety of purposes, and we have estrogen, progesterone, correct? Androgens in males. So they control the genes which we have. We have about 20,000 genes. Each steroid hormone controls a subset of genes. Some genes could be controlled by more than one hormone, correct? So that's what we study, how steroid controls the gene expression in normal cell or in disease cell. This particular topic was related to cancer. So these are some of the stuff we do. We look at a family of enzymes. These are called mixed lineage leukemia. Those are critical player in leukemia. Okay, the children, about 8 to 10% of the childhood leukemia has something to do with this particular gene mutation. And normally some of the mutations are so aggressive that nothing can be done. Okay? And anything we could do, there, doctors, parents, and they are going to try because there is no therapy except the transplantation of the stem cell, correct? Bone marrow stem cell therapy, where you remove the damaged bone marrow stem cell mutated stem cell and then replace with some good stem cell, bone marrow transplantation, where, correct, there is hope that there will be proper tissue matching and eventually help regenerating the blood cells again, healthy blood cells. So we try to understand the mechanism by which this enzyme functions, and it appears that this family enzyme actually controls the chromatin dynamics. So we have this chromosome, and this family of enzymes control the chromosomal dynamics, gene expression regulation. They're basically histone methyltransferases. We'll talk briefly about it. And in my lab, we show that this family of enzymes are actually critical players in hormone-dependent gene activation. Whenever there is a hormone, this family of gene comes into play. Okay, they interact with something called nuclear hormone receptors. So these are the proteins through which the hormone interacts. So wherever you see hormone, there is these family of enzymes are working. We have done some work on cholesterol metabolism. In fact, we just succeeded in an NIH grant where we are studying how the enzymes control the estrogen-dependent cholesterol metabolism. Okay, in cholesterol and fat metabolism, one of the organs which plays critical role is your liver. Okay, liver controls this, and liver function appears to be very closely related with the hormone. Okay, cardiovascular is lipid metabolism, and it's well known that especially in female premenopausal stage, there are much lower risk of cardiovascular disease, stroke, heart attack. It's much higher in the male, because which says that this hormone, ovarian hormone, plays certain role, correct? And when we go to the postmenopausal stage, then your ovarian hormones are not being produced as much, correct? It starts the cardiovascular risk, lipid, homeostasis, cholesterol level, all these shoots high, and the risk of, correct, cardiovascular disease also increase. In those circumstances, people normally go for hormone therapy to control, but hormone therapy is not good at all, because the external administration of hormones or hormone analogs, while it takes care of your cholesterol level to some extent, but it triggers many other factors, including cancer causing oncogene activation. Does it make sense? So hormone therapy is not a solution. There is a lot more, I don't want to go in detail. So what I am trying to say, that ovarian hormones has a significant function in maintaining the lipid and cholesterol levels, correct? So what the purpose of this NIH grant is to look how effective ovarian hormone uses the liver to control the lipid and cholesterol metabolism. They are involved in neuronal function and many others. We look at some of the cancer biology, especially on the epigenetic aspect, and because we do understand good amount of cell signaling biology, my lab actually heavily involved in developing some drugs, gene therapy or small molecule. So in prokaryotes, in eukaryotes, your DNA is there. DNA is transcribed into RNA, RNA is transcribed into protein. This is a classical picture of genetics, correct? Mendelian genetics. That your DNA has the allele, correct, which links to the phenotype. So that is a Mendelian genetics, 1816, correct? So two different persons, so different because they have different genes, their genetics are different. This is what Mendel said. I wish it was Mendel, but correct? Maybe something else. So when you talk about development of a single organism like human, you start with a single cell, becomes 2, 4, 8, 16 and so on, correct? When you have a zygote, it's a single cell. That single cell, when it divides, the genetics, the DNA has to be duplicated identically. Otherwise there is a mutation, correct? So each cell in a fully grown organism, as it comes from a single cell, their genetics is identical. But during this developmental process, at some point, some of these cells has to be differentiated into neurons, correct? Blood cells, bone cells, even if they have same genetics. And this process is controlled by the differential expression of the genes. We have 20,000 genes here, everywhere, but not all the genes are expressed all time. So certain genes are expressed, certain genes are silent at a given time, maybe at the first 3 hours of fertilization. There are critical genes which are involved in stem cell production and maintenance. Correct? Those genes are expressed. But as the cell differentiates into a particular class of cells, those stem cell type of genes are turned off. So you produce only genes which are specialized for that type of tissue. Does it make sense? So this is a whole thing of gene expression and regulation, how you can control, turn on and so on. So how this differential gene expression is controlled? What is the mechanism of regulation? There are very, very tiny amounts of hormones. Does it make sense? So the early stages of development, tiny amount of hormones actually controls this differentiation process. If there is a problem or disturbance in the amount of hormone or abnormal hormone, differentiation will not take place properly. Abnormal development, autism and many other stuff happen. So my lab looks at how these genes are turned on in the human system and so on. In eukaryotes, your DNA is double helical DNA. Now we are getting back to your study. Okay? So in eukaryote, your DNA is double helical DNA. Now they are complex with variety of proteins, basically highly complex with variety of nuclear proteins and form chromosome and very condensed form of chromosome which you see only in the mitosis. Okay, it's called chromosome. If you take the cells, let it go into mitosis, stain the chromosome with some fluorescent dye, look at under microscope, you actually can count all 46 chromosomes. But normal cells which are in the interphase or G1, G2 synthesis, you won't be able to see these chromosomes. They are dispersed in the form of nuclear matrix. Does it make sense? So if you look at these, so there are different order of chromosome organization. If you look at this, one of the simplified chromatin, it looks like that. Your DNA, which is the red ribbon here, is wrapped around certain nuclear proteins which we call histones. You studied in Biochem 1 some of these, correct? So there are 4 histone proteins, H2A, H2B, H3 and H4. So there are 4 histone proteins, H3, H4, H2A, H2B, which forms the nucleosome core. the simplest nucleosome, and this is wrapped with the DNA about 1.7 tons. 1.7 tons of the DNA around this protein. And then you have another nucleosome and so on. So this is called a single nucleosome. Many nucleosomes are formed, correct? And they further condense with the help of other proteins, modification, and eventually they form these condensed chromatin. Now, if you look at the structure of these histones, what is the charge on DNA? Negative, phosphate, correct? The histones are enriched in basic amino acids like lysine, arginine, correct? So histone has this nucleosome surface as a positive charge. Does it make sense? And that positive charge can easily form complex electrostatic with negatively charged DNA. For a long time, people thought that function of nucleosome is to complex with this negatively charged DNA polymer so that large piece of DNA could be compacted in the tiny nucleus. Does it make sense? Now, starting 1998, the field of epigenetics came in. What it says, along with this charge, this histone has a very dynamic role, crucial role. In addition to compacting DNA, it also controls the gene activity. Does it make sense? I give you an example. Let's say this is lysine. Lysine has a NH3 group, positive charge. Correct? This is, let's say, histone H3. This is a lysine at 4 position. So if this is positively charged, it binds to DNA negatively charged. So DNA is compact. Does it make sense? But there is a class of enzyme which adds an acetyl group on this lysine, okay, that forms NHCO3. It's acetylation of the lysine. What cofactor you need? You need what cofactor? You need acetyl CoA. Correct? And you need some enzymes which are called histone acetyl transferase. Also, they are called HATs, right? So there are many histone acetyl transferase. Think about this. This is lysine. Is it positively charged? So DNA is complex, correct? Now you add the acetyl group, it becomes acetyl lysine. What is the charge on the nitrogen side chain? Neutral. So it's 0. Can DNA interact now? So DNA becomes free. Does it make sense? DNA becomes covalent. So here DNA was complex. It was not free. So it is kind of repressed. Does it make sense? But when it is acetylated, it becomes open. So DNA genes which were repressed because of the acetylation, gene starts to be activated. Does it make sense? Any genes in our body, in our cells, any eukaryotic cells, whenever there is a need for transcription activation, the histone proteins must have to be acetylated so that DNA becomes open. Does it make sense? Now, there is another class of enzyme which can remove this acetyl group. Just this is a amide linkage, so you hydrolyze it, so it goes back. This is called histone deacetylases. Histone deacetylase. They are also brief, it's called HDAS. Histone deacetylase. What is the function of histone deacetylase on gene expression? Whether it will activate or repress? It will repress. Does it make sense? So basically, it's not the DNA. It's the modification on the histone protein, correct? Covalent modification on the histone protein that's actually controlling the status of the gene, whether it should be expressed or it should be repressed. I gave you one of the most simple examples. Okay, in fact, HDAC inhibitors, so histone deacetylase inhibitors are actually drugs, chemotherapeutic drugs for cancer. Many anti-cancer agents are just HDAC inhibitors. Any of you work with Dr. Frost lab? So, you know with whom Dr. Frost did his postdoc? Well, you need to know if you want to make money. Okay, Ron Breslow, and he is in Columbia University. One of the, he's not Nobel laureate, but highly respected organic chemist. And he made a molecule, the most famous, at least one of the most famous, widely used anti-cancer drugs. Okay, this is called SAHA. Okay. Anybody knows what it is? It's nothing other than a DMSO, dimethyl sulfoxide analog. What he saw that if you take the cancer cell at the DMSO, cells are dying. But the problem is that DMSO is a strong organic solvent. It kills cancer cell as well as normal cells. Does it make sense? So he thought if we can modify this molecule in such a way that you keep the cancer cell specific activity and take away the normal cell specific activity, correct? Then it could be used as drug for the treatment of cancer. And this is his discovery. It's a subbaric acid, dicarboxylic acid, based hydroxamic acid derivative, nothing other than a dimethyl sulfoxide analog. It's a widely used anti-cancer drug, and of course, the mechanism says that it's a HDAC inhibitor. Many growth hormones in nature, like auxin, gibberellic acid, I believe they are all histone deacetylase inhibitor because growth hormone stimulates the plant growth, correct? So to stimulate, you need more protein. That means you need to, correct, inhibit these HDACs so that histones are acetylated. Does it make sense? So this is how one of the mechanisms how genes are regulated. So along with histones, there is acetylation, there is methylation, phosphorylation, and many other modifications which I don't want to go into detail in this class. They control the gene activation and silencing events. Couple of years ago, there was a fine scientist who proposed that beyond the genetic code, correct? DNA linking to RNA to protein, correct? That's a gene is controlling our phenotype. Gene alone is not, gene is important, genetics is important, but gene alone cannot control everything. So gene environment, where the gene is, what hormone, what temperature, correct, is there, that dictates what the protein will be expressed, what the phenotype. Does it make sense? That means if you put human into the extreme cold, human will never become dinosaur. Correct? Genetics is totally different, correct? But if you interfere with the normal developmental process, especially the embryonic stage, development can be misguided. That contributes to autism, abnormal development, and human disease, and so on. So this chromatin modification controls the gene activity. There is a hypothesis called histone code hypothesis, that histone modification on the chromatin controls, is a kind of code which controls the gene silencing. Last year I edited a book with Wiley publisher called Gene Expression, Hormone Signaling, Epigenetics and Hormone Signaling. And there in one of the chapters, I actually hypothesized that you have genetic code and somebody did histone code. I think all these are important, but there is more to these, which we try to say epigenetic codes. Epigenetic code hypothesis, where we hypothesize that along with the genetic code, along with the histone code, there are many other factors, correct, which control this gene environment and controls the gene activation and silencing. So this is extremely complex and regulated. I think rest of the stuff you don't need to see. We do a lot of cell biology, hormone regulation, disorder. We do a lot of stuff. I want to show you one slide to make you a little bit excited. We do a lot of cancer biology. Where are those slides? Yes. So we work on these chromatins, modified, mixed lineage leukemia genes, which are involved in cancer, leukemia. Now, they are also expressed in other cells and sometimes highly expressed in human cells to see what happens, how exactly they contribute to tumor growth. This is a couple of years old slide, that's why, but it is published. So what we see, that we took different types of cancer cell, normal cell, cancer, normal, cancer, normal. This is a colon cancer cell. This is colon normal, breast cancer, breast normal, lung cancer, lung normal. We knock down one of these genes, which is MLL1. If you knock it down, you see these are human cells cultured in place. You should get into lab and try to get going some of this experience. If you knock down only one gene, you see that cells are rounded up, dying. Okay? I don't understand, but for some reason these healthy cells are not as much affected. Okay? But again, in lung cancer we see healthy as well as cancer cells, both are affected. So because it is killing the tumor cells, we did all the biochemistry and we thought, can it control the tumor growth in vivo? These are nasty pictures. Okay? So we took some mice, implanted a human tumor. So if this tumor is rapidly growing, you see it's just implanted in 28 days, it is really nasty. Okay? But in this case, we applied one of the MLL1 targeting antisense. As soon as you knock down this particular gene, you see the tumor is not growing. Does it make sense? So, what it says that this is a potential target for treating some of the cancers. So, this is the tumor growth in control. This is the tumor growth in the treated animal. Okay. It's an enzyme, and we have done a lot more work here. It controls the tumor growth factors, angiogenic factors, and so on. So, it is a potential target for therapy. I skip all the biology by cell signaling biology. Okay. So, what we show that MLL1, which is the histone methyltransferase, it's an epigenetic regulator, which is critical for normal cell growth in hematopoiesis, blood cell differentiation, is a potential target. It can be targeted to repress the tumor growth and therapy. So in my lab, we are actually screening small molecules and libraries which can inhibit this enzyme to treat some of these diseases. So many times these are extremely good ideas, good discovery, but it does not end up with a product in the patient bedside. There's a lot of things are involved, okay, including large amount of money. So, but at least we try to do some of the stuff, understanding the human biology. You see, I have a lot of undergraduates. I tell you to get into the lab. These are all undergraduates. This is an undergraduate, Dave Grant. I was the assistant professor at the time, and he somehow liked my classes back and said, "Can I work in that?" I said, "Okay." He was getting at the C class, okay, but he got so much excited. He actually ended up with 1 patent and 4 publications from my lab. Okay, right now he got PhD in neuroscience from Purdue University. So that's why I encourage you to get into the lab. Just don't, right? You have get excited and so on. I take a lot of undergraduates and not all of them are successful. Right? But some who likes to work, they have the potential. I think last undergraduate I have, she has been co-authored in a paper and she's getting at least 2 other. She's going to med school. Okay. So I encourage that all of you explore some lab at this point. It doesn't matter what you want to do, correct? At least you will get some exposure to what goes on. So with that lecture, let's see where we stand. So we are talking about eukaryotic genes regulation. Okay? So in eukaryotes, it's not like an operon. Genes are separated. For each gene, you have a promoter, correct? Introns intervene between exons. Correct? So, I think these are the concepts we discussed. So, these are introns. So these are only protein coding genes. Those are tiny, correct? Protein coding sequences. They are intervened with lots of introns. Function of introns may be favor alternative splicing, protein diversity. Now we know they actually produce a lot of non-coding RNA. Capping and cap methylation, these are critical for its regulation, correct? If mRNA is not capped, it gets degraded. So capping is a regulation. Addition of the cap, and the cap is nothing other than addition of a gene okay, guanosine, and this nitrogen gets methylated by a methyl transferase amylo enzyme. The discoverer of mRNA capping is actually Aaron Sepkin. I think 2-3 years ago he passed away, but one of the fine scientists I have ever worked with. Okay, normally scientists will be very angry, correct? Shout and all this stuff. I do sometimes. I'm not the best one, but I have never seen him losing his temper. Every time I have a problem, he will encourage me what to do, and he is actually discovered out of this mRNA capping, not the Nobel laureate, but a high profile scientist. So mRNA capping is very important. Polyadenylation 3'N, it is also a regulatory mechanism. 100 to 200 nucleotide adenine residues are added at the mRNA transcript, and this is done by polymerase. Does it make sense? We already went over this. I'm just revising. Then pre-mRNA is processed, spliced to produce the mature mRNA, and there are splice sites, which are the sequences which are recognized by splicing enzyme. What is the regulation stuff? Oh, yeah. So this is what we discussed, that you have histone proteins, DNA is wrapped around the histone protein 1.7 ton. How long is the DNA? What is the base pair per nucleosome? How many base pair long DNA? We said 1.7 ton. How long is the DNA? Per ton of per nucleosome? You studied this in Biochem 1. Correct? Nucleic acid. Anybody? 10? No, 10 is one helix. Right? Any? Somebody said. So this is about 147 base pair per nucleosome. There is a little bit DNA in between this. So total per nucleosome, including the linkers, is about 200 nucleotides. So if you have a plasmid DNA, if you deposit nucleosome, let's say plasmid is 5,000 base pair long, how many nucleosomes can you deposit in this plasmid? How many? Huh? You are right. So about 25 nucleosomes. Does it make sense? So can I ask you that how many nucleosomes you have, in your single cell? Can I ask? Yes or no? Why no? So you know how many base pairs you have, correct? 6 billion, correct? 3.3 to 10 to the 9 something, correct? So you divide by 200. Correct? So you can calculate how many nucleosomes you have per cell. Does it make sense? Now can I ask? So in reality, actually, you don't have that many nucleosomes. That's theoretical. it's much less because there are certain region of the genome is not as condensed, some region is more condensed, somewhere is more nucleosome rich, somewhere is. But normally, if you have a DNA of this length, you should be able to deposit every 200 nucleotides one nucleosome. Does it make sense? So this is called nucleosome code. These are the histone proteins. Histone proteins goes into modification. So I said histone acetylation opens at the chromatin, deacetylation, correct? Condenses the chromatin. Methylation does not do that. We are almost done. I want to introduce another concept and then go. So histone acetylation in general increases the gene expression. Let's say this is histone H3. There are a couple of lysine, A4, A9, A27, A36, A79. Length of the histones are about 110 amino acids. Methylation at lysine 4 is linked with gene activation. Methylation in lysine 9 and 27 is linked with silencing. Okay. Now what happens into the methylation? So acetylation neutralizes the charge, correct? What does methylation do? It does not. So it can add a CH3 group. Does it make sense? It can add actually another CH3 group because you still have the NH2, correct? The primary, secondary amine. Alright? So, and you have charge. When it is positively charged, it is replaced. When it is methylated, it is also positively charged. Does it make sense? So, what do you think it will happen? It will be replaced. But that does not happen all the time. Depending on which lysine is methylated, that may activate or suppress gene expression. So it's not only charge, there is something else. What happens is that when these are modified, acetylated, methylated, they act as a landing platform, docking site for other transcription factors. Does it make sense? It's like platform. The DNA is a platform, and there is certain place and correct transcription activator or replacer has to land on the platform to do the job. Does it make sense? That landing or recruitment is decided by this set of modification or code. Does it make sense? Acetylation and H3 lysine 4 methylation will recruit activators, whereas lysine 9 and 27 methylation will recruit replacer. In fact, these modifications are so critical. H3 lysine 27 methyltransferase is a biomarker for breast cancer. There are a lot of pharmaceutical companies are developing inhibitors for H3 lysine 27 methyltransferase, which are potential drugs for cancer and some other neurological. Right? So, understanding the signaling biology mechanism is very important, and imperative is highly complex, and if they are dysregulated, it causes cancer or other disease. Correct? So, with this, we close the transcription, and next class we will start translation. Next chapter. This I have already uploaded, correct? So I'll upload the configuration. If you don't get uploaded, just put me an email.